Hello everyone, welcome back to my channel Pari's tutorial. I'm back with one more interesting video. So let's get into the class. Today I want to discuss about the last separation technique or last important technique for the protein characterization and detection of mass called mass spectrometer. Okay, it is also included in the syllabus for the life sciences under post graduation preparations. Mass spectrometer is a technique which help you to decide the mass of that particular protein. See, the main basic principle involved in spectrometer is how, when in the presence of electric field, how the ions move based on their mass. What is the acceleration of that particular ion in the presence of electric field? You all know that proteins and peptide molecules are not volatile, right? They are all, you can't make them transfer into a gaseous phase, because main mass spectroscopy is mainly based on that. You have to convert those protein molecules into ions. That can only happen when they are made to stabilize or when they are made to co-precipitate on the organic compound. This organic compound should be able to absorb the light from a laser of certain frequency, where that would provoke the ions from the surface, so that the ions would travel in the presence of electric field, right? That is the main principle involved. So you can also compare that with the Newton's law. Say, what is F is equals to ma? Think that F is the electric field, that electric force or electric field that you are implying. Okay, and this is the mass that you want to detect of unknown protein, and A is the acceleration. So you can detect the movement of those particular ions under electric field can be determined based on the mass. Higher masses present in electric field would move slower than the smaller particles, which can easily move through the electric field, right? When the substance, like when the proteins or molecules are made to sit on organic compound and made to co-precipitate on it, so that the laser beam light focuses on it with certain wavelength, all the ions start distributing from your surface, and it will start moving. So when you place the electric field upon the electric field, they get attracted, go to the detector. So the small ions would obviously go faster than the larger ions. So based on it, the movement would be little lesser for the larger molecules. So we can calculate the acceleration. You can calculate the mass of that particular protein at the same electric field given. For example, if you got two different ions, these two different ions has got the same charge, but they are different. They are having two different masses. Okay, when they are placed in the electric field, the one ion which has a higher weight would obviously go slower towards the detector, but the smaller ions, being having less weight, would move a bit faster than your larger molecules towards the detector, and would be easy for the detection. So this is the whole principle involved in mass spectroscopy, and the whole thing is dependent on the type of matrix that you are choosing. Like see, that is why this mass spectroscopy is mainly relying on MALDI, what is called matrix assisted laser desorption and ionization, right? So you need a matrix for your compound to sit on. Then you will be adding a laser of certain wavelength, so that the particles get ionized. Once upon ionization in the presence of electric field, you will catch hold them in your detector, where it detects the time of flight. What is called time of flight? The time that the ions has taken, that for the movement from your base towards your detector, gives you the time of flight. How far that particular ion is traveling, it depends upon the molecular weight, or it depends upon the weight of that particular particle. Obviously, the smaller particles would move faster and get detected fast, and be getting a big graph there. In the same way, the bigger particles move slower because of its weight, so acceleration would also be lesser, so detection would be taking time for larger molecules. So this is how you detect the spectrophotometry. So I'll tell you with the diagrammatic representation. See, this is, just think, this is your matrix where your compounds or the protein of interest is placed. Okay, and when your sample is placed, and this is a tube, okay, where the proteins gets ionized and has to travel towards the detector. Okay, this ionic source you will be getting from the beam. I told you laser light will be having certain wavelength where it will give the ions, okay, of that particular protein. So the ions of bigger charge will be staying back, and ions with smaller charges and smaller weight and smaller mass would be going faster. So based on that you will detect them, and you will calculate the TOF. TOF is the time of light. How much time does that particular ion has taken to travel from the ionic source to the detector. So this is a simple functional mode of MALDI. Okay, so this is the principle. So hope you got an idea about it. So this is one way of detecting mass, spectrophotometer. At the same way, electrospray. Now we have covered the protein separation, we have covered the RNA separation, DNA separation, what are the things involved, what are the configuration. We have gone through chromatography, we have gone through blotting techniques and all. So this is the last class for your separation techniques. Hope I am clear. I hope I am. Next class I will start with the cell biology, all the types of cells, and also the important metabolic pathways that happen in mitochondria and other organelles. I will be discussing in cell biology. Okay, so see you in the next class. Thank you and have a nice day. I have attached the links for the topics in the description box. Please check it out.